Hi everyone and welcome to the channel. This video is about DNA fingerprinting. Now you probably know what a fingerprint is. We all have ridges on our fingertips that leave a unique pattern when we touch certain surfaces. This is very important in forensic science because fingerprints are markers of human identity. Fingerprints found in a crime scene can be compared with a suspect's fingerprint to identify the culprit. DNA fingerprinting is a similar technique. Oftentimes, the culprit gets injured and leaves traces of blood behind. This blood contains cells that carry DNA which can be used to create a unique barcode-like pattern, and this can be compared with the suspect's DNA to identify the culprit. So let's understand how this unique pattern is produced. Human beings share 99.9% similarity in DNA sequences. The remaining 0.1% is different in all of us. That means that out of the 3 billion sequences we all have, there are about 3 million DNA sequences which vary between any two individuals. Now, you don't need to compare all 3 million sequences to differentiate individuals. You can distinguish individuals by comparing just specific regions that contain repetitive sequences called satellite DNA. Satellite DNA is a term used to describe tandemly repeating non-coding DNA. So what does that mean? Imagine this is the DNA sequence in a single strand at a specific chromosomal locus. These 10 base pairs are being repeated tandemly. That means one after the other with no interfering sequences. Such sequences are found throughout the human genome, especially in the telomere, centromere, and the region surrounding the centromere. They were initially considered to be junk DNA because they didn't contain any information for making proteins, but was later found to play an important role in chromosome segregation during cell division. Some satellite regions have sequences that repeat the same number of times in all individuals, whereas other satellites vary in the number of repeats from individual to individual. These satellites are called VNTRs, that is variable number of tandem repeats. Their sequences about 10 to 100 base pairs long that are repeated between 5 to 50 times in the genome. Here, in both the individuals, there is a 10 base pair sequence that is being repeated tandemly. In individual A, the sequence is repeated 6 times, whereas in individual B, the sequence is repeated 5 times. This is an example of a VNTR where the number of repeats vary from one individual to the other. This variation in repeats is seen even between the homologous chromosomes of the same individual. Remember that we have 23 pairs of chromosomes. One set of chromosome is inherited from the father and the other set is inherited from the mother. As a result, the number of repeats present in the paternal and maternal chromosome also varies. This variation in genetic information can be used not just to identify individuals but even the family relationship between them. And this was realized by a scientist named Alec Jeffreys in 1984. Alec Jeffreys used the Southern blotting method to develop DNA fingerprints of individuals. Southern blotting is a method used to identify a specific DNA sequence in blood or tissue samples. This involves 7 major steps and I'm going to explain them one by one. The first step is to isolate the DNA from the cells. Every cell in our body contains the same DNA. So DNA can be extracted from various human materials like blood, hair root, saliva, etc. Once the DNA is extracted, it is fragmented using restriction enzymes. Restriction enzymes are enzymes that cut DNA at specific sites. H3 is an example of a restriction enzyme used in DNA fingerprinting. It recognizes the sequence GGCC and cut the DNA at the center of this sequence. GGCC is a sequence that can be commonly found throughout the genome, even near the VNTR region. This causes the entire genome to be cut into thousands of pieces varying in length. Also take note that the restriction enzyme is selected such that the sequence it recognizes is not found within the VNTR. This prevents the fragmentation of the VNTR. The VNTR selected for DNA fingerprinting by Alex Jeffrey was a 33 base pair repeat that is found in multiple locations containing different number of tandem repeats. This means that there are multiple fragments of DNA containing different number of the repeats. These DNA pieces can be separated according to their length using gel electrophoresis, which uses an electric current to pull the negatively charged DNA through a mesh-like gel. This allows these smaller fragments to travel faster than the larger DNA fragments. Now we have the entire human genome fragmented and arranged based on their size. But what we really need is just the DNA fragments containing a specific VNTR sequence. To visualize only the DNA of interest, a DNA probe is used. This probe is a single-stranded DNA molecule having complementary sequences to the VNTR. This means that it will bind specifically to the DNA band containing the VNTR, and it has a radioactive material attached to it for its detection. But the probe cannot access the complementary sequence in a double-stranded DNA. So first, the DNA strand has to be separated. This is called denaturation, and it is usually done by immersing the gel in sodium hydroxide solution. Even after the DNA is separated, the probe will not be able to access the DNA molecules embedded in the gel. Hence, the separated strands have to be transferred from the gel to a thin membrane made of either nitrocellulose or nylon material. This is done by placing the membrane and the gel in this setup. As you can see, the gel is kept in a tray containing a buffer. On top of the gel is the membrane onto which the DNA will be transferred. Above the membrane is a stack of paper towels, and lastly a heavy object is placed on top. Keeping this weight will ensure that there is even contact between the gel and the membrane. So the gel is placed in this setup overnight, during which the dry paper towels will draw the buffer solution through the gel and the membrane due to capillary action. The suction of the buffer through the gel and the membrane causes the DNA molecules to travel from the gel to the membrane. In other words, the DNA is blotted onto the membrane. The DNA then sticks to the membrane, retaining the pattern it had in the gel. Once the membrane is fully dry, the DNA is almost permanently immobilized. This means that the DNA fragments cannot be washed away. Next, a solution containing the DNA probe molecules can be added on top of the membrane. And finally, the probe can access the separated DNA strands on the membrane and hybridize or bind specifically to its complementary sequence. Like I said before, the probe has sequences complementary to the VNTR, so it will bind only to the DNA band containing the VNTR. The probe is allowed to bind to the DNA overnight, and the excess unbound probe is washed off. The final step is to determine which DNA bands are bound to the probe. As I said before, the probes are incorporated with a radioactive material like radioactive phosphate group. When an X-ray film is placed over the membrane, this radioisotope emits radiation that ionizes the silver halide coating on the X-ray films. This creates a silver precipitate that forms a dark band on the X-ray film. This is called autoradiography. The position of each band on the X-ray film will depend on the number of tandem repeats in the DNA fragment. These bands will form a pattern unique to an individual. Hence, this pattern can be used to identify an individual. Every individual will have a different pattern except in the case of identical twins. This is because they were separated from the same zygote and have identical genetic sequences. This pattern can also be used to establish the family relationship between individuals. Every child inherits 23 chromosomes from the father and another 23 chromosomes from the mother. This means that the VNTRs are also inherited from the parents. As a result, a person's DNA fingerprint or profile will partially match the parent's DNA profile such that 50% of the bands will match the father's profile and the remaining 50% will match the mother's profile. This makes DNA fingerprinting useful for paternity and maternity testing as well. So let me end this video with a simple exercise. Here again you have the DNA profile of the two parents. The elder daughter is actually a child from a previous marriage. So can you determine who is the biological parent of the elder daughter? Leave your answer in the comment section below. Thank you for staying till the end of the video. Subscribe to my channel to see more content like this. And if you missed my previous video, then do check it out.